Neo is facing a dilemma. He wants the red pill, but unfortunately, he has a rare genetic condition called achromatopsia. This means he can't see which pill is red because both pills appear grey. In fact, his whole world is reduced to grayscale, and he also experiences severe visual impairment, involuntary eye movements, and photophobia. But thanks to cutting-edge ocular gene therapy, scientists are working to restore colour vision in those affected. Welcome to the genetic basis of stuff and things. Achromatopsia affects about 1 in 30,000 people worldwide, but in the remote Pinch-Lap Atoll in Micronesia, about 1 in 25 people have the condition. This unusually high prevalence is due to a founder effect, after a devastating typhoon in the late 1700s left only 20 survivors, with one being a carrier for this vision defect. Vision begins when light enters the eye and hits the retina, where specialized cone photoreceptors reside. These cells are responsible for high acuity daylight vision and enable color perception. Unlike color blindness, which typically affects one type of cone cell, achromatopsia impairs all three types, each sensitive to different wavelengths of light. While tinted glasses may ease light sensitivity, they don't address color vision loss. Achromatopsia is mostly caused by a defect in the cone phototransduction cascade. That's the mechanism by which our eyes convert light into electrical signals for the brain to interpret. It is inherited in an autosomal recessive manner, which means the condition manifests when both copies of the gene are defective. In most cases, this leads to a complete lack of cone photoreceptor function from birth or early infancy, characterized by slow progressive degeneration. About 90% of mutations occur in the genes encoding the cone cyclic nucleotide gated, or CNG, channels, CNGA3 and CNGB3. These channels are essential for regulating ion flow in response to light, acting as cellular switches essential for vision. There are over 81 single nucleotide polymorphisms, or SNPs, in the CNGA3 gene that are known to influence the production of that protein. A common one is the conversion of guanine to adenine at the 1585th base. That leads to a missense mutation where it codes for methionine instead of valine at the 529th position. Researchers showed that CNGA3 was involved in achromatopsia by knocking out the gene, creating mice with the condition. They then corrected this by reintroducing the wild-type CNGA3 gene, enabling CNG channel function. They used adeno-associated viruses, known as AAVs, as gene delivery vectors, which are promising for eye-targeted therapies due to their non-pathogenic nature and ability to elicit a minimal immune response, especially in this immune-privileged environment. In animal models, AAVs efficiently transfer the therapeutic gene while rarely integrating into the host genome, reducing mutation risks. Once delivered, the CNGA3 gene is expressed, restoring CNG channel function and supporting long-term gene expression. This precision and safety make them ideal for advancing treatments from animal models to clinical applications. So what about for humans? In 2020, the first human phase 1/2 clinical trial targeting CNGA3-related achromatopsia using gene therapy was conducted by Dominic Fisher and his team. It evaluated the safety and vision outcomes of a single subretinal injection using an AAV vector to deliver a functional CNGA3 gene. The AAV vector was specifically designed to target cone cells, carrying a full-length human CNGA3 gene, controlled by a cone photoreceptor-specific human promoter. Additional elements were included to ensure full and enhanced gene expression. 9 patients with complete achromatopsia due to faulty CNGA3 genes received this therapy. After 1 year, patients demonstrated significant improvements in visual acuity, contrast sensitivity, and color vision. 5 out of 9 patients exhibited normalized pupil constrictions, a direct measure of visual acuity. Patient-reported outcomes indicated enhanced abilities in differentiating colors and identifying letters and numbers, key tasks in daily life. This therapy showed an excellent safety profile with no significant adverse effects and no detectable viral shedding, indicating that the vector remained localized in the retina, just as the researchers expected. These findings underscore the potential of gene therapy to safely improve cone function in adults with achromatopsia, offering a pathway to improving color vision and overall quality of life. It also suggests that earlier treatment when brain plasticity is higher could lead to even better outcomes. This research not only provides compelling evidence for the safety and efficacy of this approach, but also sets the stage for future studies aimed at optimizing treatment timing and strategies for more effective therapies for achromatopsia and similar retinal disorders. The 3-year follow-up confirmed the treatment's safety, but larger sample sizes are needed to fully demonstrate its efficacy before it can be widely rolled out. We are still awaiting results from 4 other clinical trials exploring gene therapies for CNGA3 and CNGB3 related achromatopsia. Ocular gene therapy is opening up a colourful future for people with inherited retinal disorders like achromatopsia. The first ocular gene therapy for retinal dystrophy has already been approved, and early clinical trial successes suggest gene therapy could offer lasting treatments or even cures, not only for achromatopsia, but also for other conditions. We're at the cusp of an exciting new era in ocular medicine.